Good morning, good afternoon, everyone. My name is Brenda Lammy. I'm Vice President of Professional Leadership Development at the college. It's my pleasure to be moderating this webinar today. As Jenny just mentioned, Q&A. If you enter your questions in the Q&A section of the webinar or of the Zoom meeting, I'll be moderating the questions at the end after the presentation. So it's my pleasure to introduce François Drolet. He's the Director of Public Affairs with Roche Diagnostics.
Thank you, Brenda, and good morning, everyone. Welcome to this webinar presented by the Canadian College of Health Leaders. It's our great pleasure to be part of this morning's presentation. As Brenda mentioned, I'm the Head of Public Affairs at Roche Diagnostics, responsible for health policy, access, and stakeholder engagement. Throughout the years, it's been now over maybe a dozen of years that we've enjoyed as an organization a very close partnership with the Canadian College of Health Leaders and its entire community. This has taken the form of many activities. You were an engaged member of the Corporate Advisory Council. We're supporting many local chapters of the college. We've set up several HPRS sessions over the years. We're the proud sponsor of the annual Mentorship Award. And we've also always provided support to the NHLC conference in the form of the Leads Breakfast and the Coach's Corner session that have been very popular over the years. So it was with very big disappointment that we received the news of the cancellation of the NHLC 2020 that was going to take place in Edmonton. Now, leadership does not take a break during a pandemic, even during a pandemic, I would say. And for that reason, I would like to take a moment to commend the college for staying at the forefront of the battle through several activities and webinars that you have presented over the past few weeks. And I would also like to thank all the members of the college who are in the trenches fighting this pandemic. The heroic work that's been accomplished by the many actors in the health system has been very critical in where we are today against the disease. Now, I would be remiss if I would not underscore the college's 50th anniversary, one that we will all remember. I'm sure I've myself turned 60 a few weeks ago, and this is now ingrained in my head for the rest of my life, I would think. But funny enough, in doing some research preparing for this this webinar, I kind of realized like some things change in 50 years, but others don't change that much. So in 1970, we used to say Premier Trudeau, and we find ourselves today saying the same thing. In 1970, was when the first Boeing 747 flight took place. And in 2020, we're sitting here maybe, trying to figure out when we will see a Boeing 747 fly again above our heads. And in 1970 also, Apollo 13 mission to moon came up with that very famous quote of Houston, we've had a problem. Well, in 2020, needless to say that we do have a problem before us as well. And since diagnostics has been at the center of the fight against COVID-19, we thought we would offer our contribution through this webinar about the patient journey. And we certainly hope that you find it useful and informative. So without any further ado, I'll introduce our speaker for today, Caroline Vincent. Caroline has completed a PhD in microbiology and next-generation sequencing at McGill University in 2015. After her PhD, she did a postdoctoral fellowship at the Quebec Public Health Lab, where she contributed the implementation of a new whole genome sequence-based method to identify Salmonella outbreaks. She then joined Roche Diagnostics Canada in 2016, and after a few other duties, currently acts as the National Medical Liaison for Cardiology pregnancy care and critical care, and now also with infectious disease. So Caroline, over to you.
Great. Thank you very much for this nice introduction, Francois. It's really a pleasure to be here with you this afternoon. So I'll start my presentation. So here are my disclaimers. And so in the first part of the presentation, I will review the main characteristics of the new coronavirus that is at the origin of this current pandemic. Then we will explore together how the illness progresses and can sometimes deteriorate in patients who contract COVID-19. And then finally, I will provide an overview of the diagnostic tests that are useful in the context of COVID-19. So, new coronavirus. So here we see the timeline of emergence of the new coronavirus. So on December 31st, 2019, this is when the World Health Organization was informed of the occurrence of a cluster of cases of pneumonia of unknown origin, which were actually related to exposure to a live animal market in Wuhan, China. Then a week later, on January 7th, the Chinese authorities sequenced the infectious agent causing this pneumonia and identified that it was caused by a new strain of coronavirus. Then at the end of January, this is when the World Health Organization declared this new coronavirus a public health emergency of international concern. Then in mid-February, the World Health Organization then announced the official name of this new virus, which is now known as SARS-CoV-2. And they also coined the official name of the disease caused by this virus, which is now well known as COVID-19. And then on March 11th, 2020, this is when the World Health Organization declared COVID-19 to be a worldwide pandemic. So just a bit on the how these virus, what are the structural features of these viruses? So basically, coronaviruses are enveloped. They are single-stranded RNA virus, so their genetic material is made of RNA rather than DNA. These viruses are known to have 4 key structural proteins. 3 of them are anchored in the envelope and are found at the surface of the virus. So these are the membrane, the envelope, and the spike protein. And then inside the envelope, we have the nucleocapsid protein, which is actually bound to the RNA and stabilizes it. So coronaviruses are zoonotic, meaning they can be transmitted between animals and humans. The animal reservoirs for these viruses are diverse, but the widest variety is actually seen in bats. And we also know that mammals can serve as intermediate hosts for these viruses, so they will serve as an intermediate between bats and humans. So there are 7 known strains of coronaviruses. 4 of them are known to be endemic, and so these strains will be responsible for approximately 5 to 10% of the common colds that we regularly see. And then the 3 other strains have now been responsible for outbreaks of severe respiratory syndrome. So we will look here more closely at these 3 outbreak strains. So the SARS-CoV-2 strain was at the origin of the severe acute respiratory syndrome outbreak. This is an outbreak that started in China in 2002. There was a little more than 8,000 confirmed cases from this outbreak, which spanned 29 different countries, including Canada. And the mortality rate was almost 10% for this outbreak. Now, this outbreak is known to have been contained since July 2003, meaning that there are no new cases that have been reported since then. Then we have the MERS, or Middle East respiratory syndrome outbreak. So the first outbreak due to the strain occurred in Saudi Arabia in 2012. And there was a second outbreak which took place in South Korea in 2015. In total, there were almost 2,500 confirmed cases with this outbreak across 27 countries. And the death rates from this infection were much higher at around 34%. And then we have the current COVID-19 pandemic, which is caused by SARS-CoV-2. And up to now, this new strain has affected 4 million people worldwide. We have now 187 countries which have reported cases of infection with this new viruses. And it has caused more than 280,000 deaths so far and continuing. So bats are thought to be the primary reservoir for all three of these outbreak strains. So in the case of the SARS-CoV strain, which caused the SARS outbreak, the intermediate host which passed the virus from bats to human was likely an animal that we call the civet cat. And in the case of the Middle East respiratory syndrome, the intermediate hosts were camels. Now, as for the current pandemic, so in the case of SARS-CoV-2, we do not currently know who is the intermediate host, but pangolins have been among the prime suspects. And then genetic analysis have shown that SARS-CoV-2 and SARS-CoV are closely related to one another, and they share about 80% sequence homology. This cartoon here depicts how the virus infects and replicates within our lung cells. So the virus has a surface protein that we call the S or spike protein. And this protein will specifically bind to a receptor that we call ACE2, and which is found at the surface of our lung cells. So this will allow the virus to attach and then enter into our host cells. And then once inside our cells, the virus will use our own host cell machinery to replicate itself and assemble new viruses that will then subsequently be released and go on to infect neighboring cells and continue the infection process. So now we're going to look more closely at the patient journey. So what happens in patient who contracts this disease? So here I will show you the main characteristics of COVID-19. First of all, we know that the virus is transmitted either directly from person to person via respiratory droplets. So either when two people talk to one another without a mask or when someone sneezes. The virus can also be transmitted indirectly via contaminated surfaces. So for instance, if a sick person touches something and then I touch it after and then I touch my face, I could contaminate myself and contract the disease indirectly this way. And then the incubation time, this is the time from initial exposure to the virus until the development of symptoms in the person who contracted the disease. In the case of COVID-19, this incubation time could be from anywhere from 2 up to 14 days, but we know that on average it's more around 5 to 6 days. And then in the next few slides, I will go into more details over the symptoms, the clinical presentation, and the progression of patients who contract the disease. And I will also talk about mortality rates. So before I jump into the specifics of COVID-19, I just want to address the question of how flu compares to COVID-19. So this slide here shows the main characteristics of these two diseases. So the R0 number, or the reproduction number of the virus, is actually a measure of the contagiousness of an infectious disease. So It basically tells the average number of people that a person with the disease is expected to infect during the course of his or her illness, if there are no preventive measures in place. So in the case of the flu, the seasonal flu, we expect that every person who contracts the flu will transmit it on average to one other person during the course of his or her illness. Now, in the case of COVID-19, the reproduction number is much higher, around 2 to 3, meaning that every person infected with SARS-CoV-2 is expected to transmit it to 2 or 3 other people on average if there are no preventative measures in place. And then if we look at the incubation time now, we also see that it is longer for COVID-19. So incubation time for flu is around 1 to 4 days, but it could be much longer in the case of COVID-19. And so this potentially gives people more time to spread the illness before they develop symptoms and actually know that they are infected. So basically, the new coronavirus transmits more easily from person to person compared to the flu. And this highlights why social distanciation measures are so important to prevent the spread of COVID-19, especially since we don't have any vaccine available for it yet. And then the early estimates right now tend to also show that the hospitalization and the mortality rates are higher for COVID-19 than the flu. Again, we have to be careful how we interpret these estimates because they are dependent on several variables, including the fraction of people who got tested for the virus and also the distribution of risk factors among the population. So these estimates should be considered provisional at this stage, but it does show a tendency towards COVID-19 to be a bit more virulent in those aspects. So now I'll show you how the illness progresses and sometimes deteriorate in people who contract COVID-19. So on the right side here of the slide, we see the clinical spectrum of the disease. So in the majority of cases, so about 80% of people who contract COVID-19, these people will either have very mild disease or pneumonia, which could be considered as a moderate severity of the disease. Then in about 14% of cases, these patients will develop severe disease, which is characterized as difficulty breathing. These are also patients who might need oxygen support. And then in a very small fraction of people, about 6% of cases, these are the patients who will go on to develop a very severe or critical form of the disease. So these patients will have respiratory failure, and then they could go on to develop septic shock, multiple organ dysfunction, and sometimes death. So here, these are the most common symptoms that patients will experience if they contract COVID-19. So these data here are based on 56,000 confirmed cases from China. We see that fever is by far the most common symptoms. It was present in 88% of the cases. And then we also typically see symptoms of dry cough and fatigue in patients who have COVID-19. And then at much lower rates, there have been cases of patients experiencing diarrhea or who lost their sense of smell and taste. So the mild cases, which, as I said, represent the majority of patients, these will typically resolve the infection after 1 to 2 weeks after symptom onset. So these are the patients who will be able to fight the disease. They won't need to be hospitalized. They can stay at home, isolate themselves, and they will eventually recover without the need for further care. Now, the more severe cases are the ones who will develop a full-blown pneumonia. So these patients will typically start to experience breathing difficulties on average 5 to 7 days after the first onset of symptoms, and they will typically need to be admitted to the hospital shortly thereafter. Now, if we look at the more severe and critical cases of COVID-19, which represent about 20% of patients diagnosed with COVID-19, these are the patients who will go on to develop acute respiratory distress syndrome. And so they will need to be admitted to the ICU, to be admitted to the intensive care unit, and potentially put under invasive mechanical ventilation at some point if their status continues to deteriorate. So the numbers that are shown here in parentheses are the proportion of hospitalized patients who experience these complications. So in the case of acute respiratory distress syndrome, so far estimates have shown that about 20 to 30% of patients go on to develop this complication during their hospitalization. And so these are the patients who will need to be admitted to the intensive care unit, and about 12 to 17% of hospitalized patients required to be intubated and put under invasive mechanical ventilation in the ICU. Oh, this shows the, here this figure shows what happens in the lungs of people who actually go on to develop this acute respiratory distress syndrome. So first, the virus will start by infecting the tiny air sacs that are found within our lungs, and which are responsible for gas exchanges between the air that we breathe and our bloodstream. So these tiny air sacs are called alveoli, and they are depicted here on this picture with the pink round bubbles. So initially, the infection will trigger a flood of immune cells in the lungs, and this will cause inflammation and vasodilatation of the blood vessels. And then in the more severe cases, what will happen is that fluid will start to build up in the lungs, and this is what will cause the breathing difficulties in these patients. And then in the most severe cases, which are depicted at the bottom of the figure, this is when the immune reaction will actually start to destroy the alveoli up to the point where they will collapse. And this is what will cause the acute respiratory distress syndrome. And as a further complication, there are cases where the fluid that has accumulated in the alveoli might subsequently spill into the bloodstream of the patient. And so this will cause sepsis, and it might eventually lead to multiple organ failure, septic shock, and death in these patients. So there's a subset of the severe cases of COVID-19 who might develop what we call a cytokine storm. So you may have heard about this. This basically occurs when there's a large number of white blood cells that are activated by the immune system, and it will lead to an overproduction of inflammatory molecules called cytokine. And these will cause the pulmonary damage and the respiratory failure in these patients. So interleukin-6 or IL-6 is one of these cytokines, and recent studies have demonstrated the clinical utility of IL-6 as a biomarker that could potentially be used for disease monitoring in COVID-19 patients. So the idea would be to measure this biomarker when the patients are admitted to the hospital in order to help predict whether these patients might deteriorate or whether they will do better and might not need to go to the ICU. So in the end, the patients who were hospitalized will either survive and be discharged from the hospital, or they will succumb from the infection. So in non-survivors, what has been observed is that death occurs after an average of 18 to 19 days after the initial onset of symptoms. The most common complications that were observed in these patients were sepsis, respiratory failure, acute respiratory distress syndrome, and septic shock. And then in the survivors, these patients were shown to have stayed in the hospital on average for 10 to 12 days. And they were typically discharged from the hospital and sent back home after an average of 22 days after the illness onset. So the mortality rates that are associated with COVID-19 have been shown to vary by country. They were around 7% in Italy, around 2% in China. In Canada, the most recent estimates are around 6.9%. It is important to keep in mind that there are many factors that affect these mortality rates. And such measures are known to be less accurate in the setting of an outbreak. I mean, among other things, this measure is dependent, as I mentioned previously, on the total number of people who are tested for the virus. There might be a significant proportion of the population who are either asymptomatic or have very mild symptoms, and if these people are not screened, then we never know that they were infected, and this might artificially increase the mortality rates that we currently see. And so, as I said, testing has been typically limited to the most sick people. These estimates are also dependent on the risk factors among the population. So if the population is older, we would expect the mortality rates to be higher. However, what we do see from this table here is that there is a clear trend towards higher mortality rates in people who are older. So we see that in the people in the age groups below 60 years old, there's less than 1% of people who will die from COVID-19. However, if we look at the people who are aged between 70 to 80 years old or above 80 years old, this proportion quickly rises to either anywhere from 8 to 20% mortality rate. So the vast majority of people who die from COVID-19 were also shown to have underlying comorbidities. Here, this is, again, data that comes from China and Italy, and where we basically see that more than 96% of people who died had at least one comorbidity. And the most common comorbidities that were observed were heart disease, diabetes, chronic respiratory disease, as well as hypertension. So now we're going to talk a bit more about diagnostic tests. So I will show you a quick video that features the General Director of the World Health Organization, Dr. Tedros Adhanom Ghebreyesus, which actually eloquently illustrates the critical importance of diagnostic testing in our fight against SARS-CoV-2. So let me load this video for you.
You cannot fight a fire blindfolded, and we cannot stop this pandemic if we don't know who is infected. We have a simple message for all countries: test, test, test.
So test, test, test. This is a clear message here. And so before I jump more deeply into the subject of diagnostic testing, we need to first recognize what are the key players in our fight against infectious diseases? The first key player is the pathogen. So in this case, it is the new coronavirus, or SARS-CoV-2, which is considered as the invader. So the second key player is the antibodies that are produced by our immune system. So that's our troopers, or what we could consider the spearhead of resistance. And then we have the general host reaction. For example, the inflammatory response of our body, which is kind of our army logistic. So we will see in the next few slides how these three components are used for diagnostic purposes. So before jumping into the topic of diagnostic tests, we first need to understand the viral infection process. So an individual will first get exposed to the virus and become infected, as I mentioned previously, either via close contact with an infected individual or indirectly by touching a contaminated surface or object and subsequently touching their face. The virus will then enter the airways and start to reproduce. So at this stage, this is what we consider the active infection stage. And so this is when people will typically develop symptoms and become infectious, meaning that they are able to transmit the disease to other people. It has been shown that viral shedding can occur for an average of 20 days after symptom onset. And as a result of the infections, patients will eventually develop specific antibodies against the virus that will be produced into the bloodstream. This is what we call seroconversion. And then eventually, people who got the infection will clear the infection and recover from it. So when it comes to diagnostic testing for infectious diseases, there are two main categories of tests available. So first we have the molecular tests. These tests are meant to identify the presence of the virus in symptomatic individuals. So they work by either detecting the nucleic acid of the virus. So this is what we call the nucleic acid amplification tests, or PCR tests. And these are really, the PCR tests are known to be the gold standard. Or they could also work by detecting the structural protein at the surface of the virus. And so these are called the antigen tests. So these tests are really made to determine whether a person is actively infected by the virus at the moment when we perform the test. And so in order to perform these tests, we will use either a nasopharyngeal or an oropharyngeal swab, although now there's work being done on the development of tests based on saliva samples, which might be easier to collect. And then the second type of tests are the serology tests. So these tests identify the presence of a host immune response against the virus by detecting the presence of antibodies that are targeting specifically the virus. So this type of test is useful to identify people who have previously been infected with the virus. And in order to perform these tests, we use a blood sample. So this figure here shows when molecular and serological tests are useful during the course of the infection. So in the early phase of the infection, when symptoms develop, this is when we can detect the presence of the virus, either by PCR or through antigen testing. So as I said, this will tell us if a person is having an active infection, and if it's the case, then it will mean that the person might be able to transmit the virus to other people and spread the infection. And then following the infection, the patients will typically develop an immune response to the virus. and they will produce antibodies that can be detected on average 10 to 14 days after symptom onset. And there are two main types of antibodies that we will be detecting. First, we have the so-called IgMs, which are shown in red here. So these tend to appear a bit later after the infection, but they do not typically stay for very long. And then we have the IgG antibodies, which are shown in orange here. And so these tend to rise a bit later, but they will stay longer than the IgMs. And this is usually this type of antibodies, so the IgGs, that will confer a certain level of immunity for many of the viral infections. So this is the antibodies that, when they mature, they will have the ability to neutralize the virus and prevent an infection. So at this point in time, there are many questions around COVID-19 and SARS-CoV-2 that still remain open. For example, we don't know yet for how long a patient remains infective and is therefore at risk of transmitting the virus to others after the person contracts the infection. We also don't know yet what proportion of cases are asymptomatic, but hopefully serology tests will be able to help answer that question. And then we don't know if the antibodies that are produced as a result of the infection confer protective immunity, and also for how long this immunity might last. So studies are currently underway to address these questions, so we should be able to get a better understanding of the disease as scientific evidence continues to accumulate. So in the context of diagnostic testing, the features that are of critical importance are the specificity and the sensitivity of the test that we will use. So the slide here highlights what a false test result would mean for the patient and what is the associated medical risk. So molecular tests such as PCR and antigen assays need to have a high sensitivity. So this means that we want them to reliably detect the presence of the virus and to minimize false negative results, because these missed cases will lead to a high risk for the infection patients who will not know that he is infected, and it will also bear a high risk of spreading the infection to other people if that person doesn't have confirmation that he or she is infected with the virus. On the other hand, when we're talking about antibody assays, we want to maximize the specificity of these tests. So we want these tests basically to be very precise in telling that a result is truly positive, because in this case, false positive results will lead to an erroneous assumption of recovery and potential immunity, which will put the person at risk of infection if she thinks or he thinks he has the presence of antibodies when it's not the case. So test accuracy is of utmost importance, and the UK government learned it the hard way last month when they bought 3.5 million antibody tests from China and later found that these tests were not reliable enough to warrant their use in the population. So the test that they bought was actually based on a fingerprint. So it was meant to detect the presence of antibodies in those finger prick blood samples. And the idea was that it would facilitate the use of these test devices at home by the patients themselves. It was already known that those finger prick based antibody tests are rarely reliable. But the recommendations from the scientists to rather focus on centralized laboratory tests were ignored. The downside of laboratory-based method is that we have to collect a blood sample in a tube that will then be sent to the central laboratory for testing. So it does take a bit more time before we get the result, but at least we know that these results will be much more accurate. So Health Canada here is the regulatory instance that is in charge of reviewing and authorizing the use of drugs and medical devices in Canada. So to address the current pandemic with COVID-19, Health Canada has put in place an expedited review process that they call the interim order, whereby they review all COVID-19 related test submissions as quickly as possible without compromising patient safety. And so through this process, Health Canada ensures that the tests are well supported by evidence and that they provide accurate and reliable results. So this is available on the Health Canada website, and we can see that currently there are 17 PCR-based tests that have been authorized for use in Canada under the COVID-19 interim order. The Roche cobas SARS-CoV-2 test is one of them. And so far at this point in time, there is no antigen-based test or serology test that have been authorized by Health Canada. But I can let you know that Roche's antibody test, which recently obtained the emergency use authorization by the FDA, has been submitted and is currently under review by Health Canada. So another key question that diagnostic tests might help us answer is basically who is Boris and who is Tom? So in other words, who will need intensive care like Boris Johnson did, and who will have a self-limited, mild form of the disease like Tom Hanks did? And so we might be able to answer this question by looking more closely at the host response to the viral infection. So this picture here shows the clinical stages of COVID-19. So we have the early infection and then pulmonary phase, followed by a hyperinflammation phase that occurs in the most severe cases. So the majority of infected people, which represents 75% of cases, will have only mild symptoms, and they will typically resolve the infection without going further than stage 1. However, in about 20% of cases, the disease will progress to a more severe form when the virus descends into the lungs, and these are the patients who will need to be admitted to the hospital. And then we have a small fraction of the cases, so about 5%, who will further progress to a critical form of the disease. And so this is what we will call the hyperinflammation phase, and those patients will be the ones who are admitted to the intensive care unit. So host biomarkers such as IL-6 or interleukin-6 could be the key here to predict early on who will deteriorate and help identify and manage these patients more earlier. Currently, there is no vaccine and there are no specific therapies available for COVID-19, so the management of the disease is really limited to the alleviation of symptoms. So for example, to provide oxygen support in those who have breathing difficulties. However, there are a number of therapeutic drugs that are currently under investigation. Remdesivir is one of them, and recently we've seen the results from a phase 3 clinical trial that looked at hospitalized patients with severe COVID-19, but unfortunately the results did not demonstrate a statistically significant clinical benefit over placebo. We also have Actemra, which is known also under the name, the generic name of tocilizumab. This drug is also being evaluated for the treatment of severe COVID-19. And there's a phase 3 clinical trial that is underway as we speak. This drug was originally approved for the treatment of rheumatoid arthritis, and it works by blocking the IL-6 production pathway, and so preventing basically the effects of the cytokine storm. So that concludes my presentation. Thank you very much for your attention, and I will now be ready to take your questions.
Wonderful. Merci beaucoup, Carolyn. That was wonderful.
Thank you.
One of the comments that came through, I think, explained it very well. I'll just clear up, and it said, excellent presentation, so easy to understand. And translating something that's so complicated for us is a skill in and of it itself. And I just wanted to mention also, Francois, merci for your introduction. It was very kind. And I also want to express, you know, the colleges and Roche Diagnostics relationship is more than a partnership. It's a collaboration, and Roche has always been there for us when we need them. So thank you for that.
Great, that's wonderful to hear.
We have some questions that were submitted prior to, well, as people registered. So I'm going to dig into those ones first, and bear with me, Carolyn, as I read through them. So first one, has it been confirmed that once you've been exposed or had the virus, you now have some immunity to the virus? I think you touched on this in your presentation, but just to emphasize your answer.
Absolutely, yes. So this is one of the key questions to which we don't have the answer yet. I mean, I know of one study that has been published, but one study is not enough to draw a conclusion, as we know. So there has been this one study showing that there are what we call those neutralizing antibodies that are produced as a result of the infection. But we don't know how long this will last. So right now, all of the so-called serology tests that are available on the market, they only tell whether antibodies are present. They don't say whether these antibodies can actually stop a recurrence of the infection. And even if they did, it's going to take time before we know how long they are protective. So these tests only say, they can only tell whether you have been previously exposed to the virus. It doesn't say, I mean, I would prefer to have antibodies than to not have antibodies if I had the choice. I mean, because having antibodies, you know, gives presumably immunity, but that remains to be confirmed with specific assays that can be done to confirm the ability of the antibodies to actually stop the infection.
Thank you. Lots to learn still.
Yes.
So.
Yes.
How can evolving viruses be identified earlier and effective vaccines developed much more quickly?
Yes, so that's a very key question. So I don't think anybody would have predicted that the next pandemic would have been caused by a new strain of coronavirus. I think a lot of the people would have suspected maybe an influenza strain to be the cause of another pandemic, because these strains are known to mutate at a bit of a faster rate than coronaviruses do. So I think that's the key question, and nobody could predict it. And then the issue is that we cannot develop a vaccine before we know what the pathogen that we're facing is. And so this is how it happens.
I quote Dr. Butler-Jones from last week often when he said, you know, Theresa Tam says, if you've been through one pandemic, you've been through one pandemic.
Right, they're not linked, and you know, it's, you know, in the case of the flu, it's a bit of a different story. I mean, what they do to develop vaccine against the flu is that they look at the strains that are circulating, and they're trying to predict which ones are going to be prevalent for the next flu season. And so they do develop vaccine based on those, sort of prediction of what strains will be circulating. Sometimes it works. They are able to have vaccines that are effective, but there's been instances where they were not able to accurately predict which strain would circulate in the next season, and the vaccines become less effective for a given season. So that's how it works more for the flu. But for the coronaviruses, so far, I mean, there had been, even for the previous outbreaks, because these were contained outbreaks and they had been limited in scope, in the end, there were no vaccines that had been developed for these coronaviruses strain.
Mm-hmm. The next question is about effectively leading a long-term care organization. Now, we had a webinar last week with Karen Bailey, who's a member of the college, and she's the CEO of a long-term care facility. And that webinar is available on our e-learning library. And I was texting with Amy in between while you were speaking, Carolyn, and we're going to email the link to our e-learning library. So for those kinds of questions, if somebody wants to access Karen Bailey's webinar from last week, they're able to do that as well. And Carolyn, you're welcome to make any suggestions or thoughts you might have on that as well.
So the question was more around how to manage patients in long-term care facilities?
Yeah, to lead a long-term care organization, yeah.
Yeah, I know that, and this is more like a society issue. So I've been following a bit what happened in Quebec. I think one of the main issues that occurred here is that basically the staff flew away. So I think, they flew away because they were afraid they would get the virus. So I'm thinking, it's all about personal protective equipment to early isolate the cases that are detected, put infected patients in a certain area, keep the uninfected patients in another area. So this is certainly a very, very hard question. And my hope is that, we will be able to learn from the current pandemic and hopefully be better prepared if, either for the next wave that might happen in theory or, similar situations that might occur in the future.
Mm-hmm. Thank you. One question that came through during the webinar was, we've been hearing talks about findings that incubation could be months, unlike what was thought originally to be 2 to 14 days. Is this true? And this,
So, yeah. So I'm not sure if we should talk about an incubation period. I think what might be happening is this question about the asymptomatic cases. So it's been estimated, and this remains to be confirmed. I mean, the serology test will tell us more accurately what is the true fraction of people who were exposed to the virus. And then there might be a significant fraction of them who never developed symptoms or very little symptoms. Right now, the estimates say that the cases that we actually reported would represent 20% of the actual cases, and there would be a hidden 80% who had very little symptoms. And so basically, when it comes to carrying the viruses for months, I don't know whether those asymptomatic cases could have the virus and carry it for so long. My assumption, and it's really my personal take on it, is that patients who are asymptomatic, they don't develop symptoms because they were good at clearing the infection. What I've seen, on the other hand, is reports of people who did develop symptoms, were hospitalized, and I've seen one report saying that viral shedding, so when they would test serially for the virus over time in these hospitalized patients, there would be virus detected for up to 37 days post-symptom onset. So I think in this case, we're talking about patients who develop more severe disease and have a hard time to clear the infection.
Mm-hmm. Okay, thank you. Wondering about another question that came in as people registered. How do we manage situations where after treatment, family members don't want, presumably the person who is post-COVID, don't want them back in their home due to fear of being contagious?
So I can respond to that question more from a diagnostic point of view. I know that the sort of the reference test that has been used so far to confirm that a patient has recovered from the infection would be to do two molecular tests or PCR-based tests within 24 hours. And if both of these tests came back negative, then the patient would be assumed to have cleared the infection because we're no longer detecting the presence of the virus in the patients, and we sort of repeated the test to confirm that it was the case. So from a laboratory standpoint, that would be how people have proceeded. Now, that being said, I am not sure, I know that testing has been limited so far to only symptomatic patients, and the number of tests were at some point limited, so we were focusing on people with certain criteria. So I'm not sure that this test has been offered, to confirm that people had recovered. So I guess, the people who are recovered are the people that, public health has followed up on, calling them to say, do you still have symptoms? And if no, then you're considered recovered. Now, for patients going back home, I think it's all a matter of, applying the basic rules of, if it's your partner, don't sleep in the same bed for a little while, wash your hands frequently, you might want to wear a mask. And so these are basic methods, knowing that this virus is transmitted via respiratory droplet. There's these basic measures that can be used to prevent the transmission if we're afraid of transmission, basically.
Great, thank you. I'm looking at the time and it looks like we're out of time, unfortunately. I could certainly listen to you all afternoon. You have a very natural skill at making this understandable, so thank you for doing that.
Great, thank you very much, and it's great to hear that, and thank you very much for having me today.
Thank you, and thanks for sharing your time. Thank you, Francois. I want to mention on behalf of the college, in addition, we have for college members, we've got groups coaching sessions Tuesday and Wednesday afternoons. They're free, they're virtual, you just need to register in advance. They're facilitated by our certified leads coaches. And also coming up on May 20th, we have a webinar called Empathetic Leadership, Caring for the Mental Health of Our Teams and Ourselves, presented by Dr. Peter Selby, who's the Chief of Medicine at Center for Addictions and Mental Health. So those are some of the upcoming webinars. Again, Carolyn, Francois, Roche Diagnostics, thank you so much for your time and contributions today. Very much appreciated.
Thanks to you. Have a wonderful afternoon.
Thank you.
Bye-bye, everyone.
Bye.